Good morning everyone. Welcome back to my channel Pari's Tutorial. I'm back with one more interesting video. Please go to the class. Good morning one and all. Welcome back to my class. In the last class I've already discussed about the structure of cell, prokaryotic cell, their function and the organelles present in it. In this class I want to discuss about the eukaryotic cell and their structure and their functions. So you all know that eukaryotes have got a well-defined nucleus with a nuclear membrane and well-defined DNA with histone proteins. Apart from these, you have also well-developed organelles present in eukaryotes, right? The eukaryotic cells are of mainly two types, like based on the presence of cell wall and without cell wall. I am separating these two into plant cell and animal cell. Plant cell has got a well-defined cell wall, whereas the animal cell doesn't have any particular cell wall. Okay, apart from those things, we have many differences in both plant cells and animal cells. So today in this class, let's discuss about the eukaryotes. Eukaryotic cells have got a very true membrane and membranous organelles. The cytoskeleton is mainly composed of the microtubules, microfilaments and intermediate filaments, which give eukaryotics a shape to it. Okay, it also helps for the intracellular movement and transport of materials. Okay, so what are the cell organelles that are present in eukaryotes? So first comes the nucleus. As I told you, the DNA is completely housed in nucleus, well defined nucleus. The nucleolus is present in the nucleus, which may be one or two in nucleus. Okay, it's a dark thing that is present where it is involved in the ribosomal formation. In the last previous class itself, you know that ribosomes are mainly made of rRNA, ribosomal RNA and proteins. So the whole ribosomal formation takes place in nucleolus. Okay, so it acts as a factory for creating the ribosomes. So next comes the nuclear envelope. Nuclear envelope is double layered membrane that protects the nucleolus and the nucleus. It is generally continued as the smooth endoplasmic reticulum and also rough endoplasmic reticulum. Okay, I will tell you later. The peroxisomes are the other organelles that are meant for the breakdown of fatty acids into hydrogen peroxide, water and oxygen. Okay, and plasma membrane, I told you, it is that protects all the organelles and it controls the molecular entry and exit. Okay, so ribosomes, as I've already told you about the ribosomes are ATS in eukaryotes. Okay, they are ATS and it has a subunit of 60S and 40S based on the sedimentation coefficient, right? So you all know that ribosomes in eukaryotes are of mainly two types. Some are ribosomes in the free cytoplasm and some are attached with the endoplasmic reticulum. So they are called as rough endoplasmic reticulum. Okay, the ribosomes that are freely in cytoplasm would help you for the protein synthesis and translation. Even the ribosomes that are present on rough endoplasmic reticulum are also involved in protein synthesis, but the polypeptide chain that is produced through the ribosomes of RER are for the only lumen proteins, are coding for only for the lumen proteins. Okay, the plasmic reticulum and smooth endoplasmic reticulum is meant for the breakdown of carbohydrates. It creates the lipids that are needed for the cell. It also helps in maintaining the calcium levels in the muscles. Okay, so secretion of exocytosis, toxins that are produced are also have to be removed out and released along with the process called exocytosis. Okay, the next comes the Golgi apparatus. So Golgi complex is a complex where you will see a stack of sac-like structures. They are like 8 to 10 present in number. Okay, based on it, you will be having two polarities. Like so, you have cis, which is forming phase, and you have trans, which is a matured phase. Okay, so these both polarity maintained Golgi complex helpful for the transport of substances from cis to trans. So based on it, the enzyme production or the enzyme maintenance or the vesicle formation, the structure vesicles for the transport of materials from one cell to other cell or one organelle to other organ is meant for the Golgi apparatus. It would pack all the materials and help in transport from cis to trans in the form of vesicles. Okay, so the cis, mostly the cis is associated with the rough endoplasmic reticulum, and the protein molecules or the glycoproteins come from the RER that would get transported to the trans. Okay, from the cis, as they are associated with rough endoplasmic reticulum, the glycoproteins that are produced here would be transported to trans form of your Golgi apparatus. That is how you that is how the Golgi apparatus work. Okay, the shipping center of proteins, I can say, they act as shipping center where it will pack all the proteins from the ER and it transports to the other parts of your cell like organelles. Okay, for example, if you want the lysosomal proteins to be transported through the Golgi, this Golgi apparatus would help you for the chemical modification of your proteins also. Like if you like lysosomal proteins need to be added with phosphates for the mannose sugars present in it, so that that particular protein would be translocated only towards your lysosome. So that is how the protein modifications or glycoprotein modifications would help. That is how the Golgi apparatus would transfer the proteins from the ER to the transform of your Golgi apparatus. Okay, so what are the lysosomes? I told you these are the membrane bound organelles in animal cells. Okay, they contain the hydrolytic enzymes that would take all the toxic materials or the unwanted molecules, so that it would be though that the cell should never get damaged, right? So it will engulf all the bad things and without disturbing the hydrolytic enzymes present in their bodies. Okay, so it gets the enzymes from the rough endoplasmic reticulum. Okay, these lysosomes work better in acidic environment. Okay, I told you lysosomes would either engulf the stuff coming from outside the cell called as endocytosis, or it will engulf the stuff that is present within the cell called autophagus with the help of phagosomes. Right? So these are the endosomes and phagosomes forming a complex called phagolysosome, where the foreign particle is completely digested, right? Okay. So apart from all these organelles, mitochondria plays a very important role in eukaryotic cells. As you know that mitochondria is meant for metabolic activities to be done. So it is called the powerhouse of cell. Whatever the energy that we get is mainly from the production of ATP. So adenosine triphosphate. This is the currency of your energy, ATP. And apart from ATP, you also got NADH, FADH2. I will discuss this in the next class itself about the metabolic pathways, how the electron transport works, how the glycolysis works. So there are many questions raised regarding this cell path. Okay, as of now I want to concentrate just on mitochondria. So mitochondria, I told you it has got outer membrane and inner membrane, and mitochondria has also got its own DNA. Okay, so it can replicate by itself. Okay, there are many diseases based on DNA mutations in mitochondria also. Okay, so generally, so mitochondria is a very important organelle. Okay, so mitochondria is mainly bound by two membranes, that is outer membrane and inner membrane. The outer membrane the inner membrane has got many foldings called as cristae. Okay, and this would increase the surface area. Okay, it has, due to which it has also got some F1 particles which are just like a stalk particles, just look like a stalk. Okay, these F1 particles are mainly important for your electron transport, where all the NADH and FADH would be converted into ATP. Okay, so the mitochondrial DNA is generally it's a closed circle DNA which resembles the bacterial DNA. Okay, so I told you the inner mitochondrial membrane possesses different lipids, enzymes and electron carriers involved in electron transport and oxidative phosphorylation. Okay, the enzymes of the tricarboxylic acid and beta oxidation pathway for the fatty acids are located in the matrix. I told you matrix is present in the inner mitochondrial membrane. It has got many different enzymes because there are many cycles going on. The TCA cycle or the oxidative phosphorylation or the electron transport would happen in mitochondria. Okay, I told you the inner surface contains F1 particles which are helpful for the ATP production. So it also uses the DNA and ribosomes to synthesize their own proteins. I told you mitochondria has got their own DNA, so they are able to synthesize their own proteins. So what is chloroplast? So this is present in the plant cell. You know that plant cell utilizes the sun energy or light energy for the production of your carbohydrate, whatever the food particles they use that, and they have to trap it. So there are two different reactions called light reaction and dark reaction that would happen in chloroplast. Okay, so these chloroplasts are nothing but the plastids. So plastids are the cytoplasmic organelles of algae and higher plants that often possess some pigments. I told you chlorophylls are chlorophyll A, chlorophyll B. There are types of chlorophylls and carotenoids. These are all the colored plastic substances that are present in the cytoplasmic of many algae and higher plants. Okay, and they are also called the storage for the food reservation. Okay, the most important type of plastid is the chloroplast. Okay, what happens in chloroplast is they mainly contains chlorophyll, which is which helps the light energy to be converted into carbon dioxide and water and to carbohydrates. Okay, it again, this chloroplast is like a mitochondria with a double membrane. Okay, outer membrane and inner membrane called the stroma. The stroma contains main thylakoids, grana and all. So the stroma lies within the membrane and contains the DNA. I told you it has same resemblance with mitochondria. It also got its own DNA or ribosomes and lipid droplets. Okay, starch granules and a complex internal membrane. So stacks of thylakoids called grana are also present in the stroma lamella. The photosynthetic reactions are generally separate in chloroplast. Chloroplast, within the chloroplast itself, there are many regions where you come across the light reaction and the dark reaction. The formation of carbohydrate from the CO2 takes place in stroma, and the trapping of light energy and to generate ATP and NADPH and oxygen, which is called a light reaction, that occurs in the thylakoid membrane. Okay, this is the structure of your chloroplast. This is the outer membrane. These are the stacks of thylakoid, and where this is where your light reaction happen. Okay, and this is the intermembrane space. The formation of carbohydrates from CO2 and water. The dark reactions takes place in stroma. Okay, this is where dark reaction takes place, and this is where light reaction takes place. Okay. Okay. Apart from all these organelles, we also got centrioles, which works on the organization of the cell by spacing where the nucleus is compared to other organelles. Okay. We also called the chromatin. I told you there are two types of chromatin: heterochromatin and euchromatin. Okay, based on the microscopic observation of euchromatin and heterochromatin, euchromatin is a light colored chromatin. Okay, and hetero is a dark colored chromatin under microscope. Okay, so cytosol, the liquid that I told you, this is cytoskeleton. The most common function is act to, as the name suggests, a skeleton for the cell to provide support and shape. Microtubules helps in transport and also helps in the support as well as the movement of the cell. Okay, so this is the rough functional table about all the things. Okay, this is please go through the slides. Even I have attached the links regarding the eukaryotic cell and their functions in the description box. Please check it out. Okay, so these are the cell organelles and their function. So this is the diagram that I want to show you: rough endoplasmic reticulum, ribosomes, nucleolus, nucleus, Golgi apparatus, vacuole. Vacuole is meant for the buoyancy. Okay, plasma membrane, mitochondria, cell wall, and all. And this animal cell is without a cell wall, so it has got much more internal cytoplasmic complex, which because of the organelles, more and more of organelles. So microtubular site. Okay, I told you these are the this is a copy of my Prescott book of microbiology. So this is the Prescott book that I followed for it. So even I will also get you the detailed books that are related for the topics. So I knew the comparison of prokaryotic and eukaryotic cell. So I've given you a shot of it, just screenshot of it, so that you can understand it. Okay, these are the differences between the prokaryotes and eukaryotes. I have covered all the organelles in brief. Okay, in the next class I will talk about the metabolic pathways and all, what happens in the organelles of a cell, what is cellular respiration, and how ATPs are generated. Okay, so hope you like the class. See you in the next class. Please subscribe, like and comment about this class in the comment box. So thank you and have a nice day.